All right, let's talk HIV and what's inside. So, if you had the world's smallest little dissecting table and a little teeny tiny scalpel and you could open up an HIV, here are some of the things that you would find inside. So, there are going to be those three components from the pro-Pol region. They all make it into the virion, which is rare for positive strand viruses. Most commonly, positive strand viruses will not package their polymerase. But retroviruses do it the retrovirus way, and so they definitely package everything. Yeah. Let's see. There is the three parts of the gag also make it in here. So the matrix, the capsid, and the nucleocapsid. And you also get a lot of those little antiviral, or rather anti-host countermeasures. Things like VPU, VIF, VPR, NEF, REV, and TAT. And so those all end up tucked away inside the virion. It's a very maximalist positive strand virus. Usually a positive strand virus is just going to rock up and translate whatever it needs because, hey, it's positive strand. It can do that, yeah. But not HIV. HIV plans ahead, yeah. It packs a big suitcase. Just one more annoying thing about HIV, I think. And then the only other major thing that the virus has made that gets onto the capsid is the envelope protein, which has that surface component and that transmembrane component. And so the surface component is also called GP120. The transmembrane is also called GP41. In HIV-1, these are the names that you can use, and so probably I will use those most commonly. But for other retroviruses, so GP120 means a glycoprotein that's exactly 120 kilodaltons. And they vary slightly in size. So you can't really call it a GP120 from another virus because it's not 120. You got to weigh 120 to be counted. Sorry. Yeah. Hence the SU and the TM. But they mean the same thing. All right. Now let's break this down a little bit further. So this is a map of the envelope protein. So from the start of the protein, which is called the amino terminus or N terminus, to the end of the protein, which is called the carboxyl terminus, this is the order of all the features. So let's have a look at them. First off, let's look at the two cleavages. So HIV actually gets cut two different times in two places by two different enzymes, and they're both necessary cuts. First one is a cut by a thing called the signal peptidase. And so a lot of proteins that are going to make their way up to the cell surface or be excreted from the cell, secreted from the cell, are going to have a thing on the front, which is called a signal peptide. And it's a signal that we need to move this protein and get it glycosylated, because pretty much everything outside the cell is glycosylated. And we also need to get it on the transports. It's got to go through the Golgi apparatus. We got to trim those glycans back. And then from the Golgi, it's got to get exported right up to the cell surface. So, yeah. The signal peptide is actually going to be cut off during the translation process. Basically, as soon as it's made, it gets chopped off, but that shunts the protein. And so now it is marked that it is going to be going to the cell surface. Next feature are these little, they look like little pitchforks, and those are going to be glycosylation sites. So these are where chains of sugar molecules are added to the protein. And these are all going to be N-linked glycosylation sites on HIV. And the N-linked glycosylation means that the glycan, the sugar molecule, is attached to an asparagine, ASPARAGINE, amino acid, which has a single letter abbreviation of N. So, and actually within the asparagine, I believe it is attached to a nitrogen, which also has the abbreviation of N. So, you know, it works either way. Doesn't matter how you remember it. N-linked glycans. Yeah. And then down here in the middle, you have a second cleavage. This is the little upside down F, and that is a thing called a furin-like cleavage, which, if you've learned anything about Other viruses like SARS coronavirus 2, that also has a furin cleavage site, as do a lot of coronaviruses. Influenza virus has one of these things. Arena viruses have them. I think Ebola might even have one. Yeah, it's a pretty common feature that you would find in viral envelope proteins, like the equivalent of this protein. Next, you see that the bulk of GP120 is going to be broken up into conserved and variable regions. And you can call these conserved or constant. Either one would mean the same thing. The conserved regions basically form the structure. They form the backbone of this thing. The variable regions in general form loops, little floppy bits on the surface. These are the bits that get changed during HIV evolution. And generally changes to V1, V2, V4, and V5 do not have much effect on the virus, but really confuse antibodies that would be binding them. They don't confuse the antibodies. It's just the antibodies don't work after you mutate these regions. But the virus does not pay a very big fitness cost for making changes in those variable regions, which is why that's where the variation goes. But the variable loop 3 is a little bit special. So there are parts of it that are super variable, but there are also parts of it that have to be pretty much intact. And they're going to play a very important function in entry. So this is a part of the GP120 that's actually going to reach into another protein, and it's going to kind of poke a little button down at the bottom of that protein. So this is going to interact directly with a protein called CCR5, and we will get to that in so much detail in just a minute. Okay, so GP120 and GP41, these two components both get incorporated onto the virion. Together they're called GP160 because I guess we're rounding GP120 plus GP41. This is going to have another several interesting regions. So at the very front, at the N-terminus of GP41, because remember, it actually gets cut with furin right here, you're going to have a thing called the fusion peptide. And the fusion peptide is going to be a little hydrophobic hook that sinks into the cell and helps the virus to reel itself into the cell and basically crash into the cell and cause the infection to happen. This is going to be helped out by HR1 and HR2, which HR1 and 2 stand for heptad repeats. And a heptad just means a group of 7 things. And so these are named after a repeating pattern where there are hydrophobic amino acids at the 1st and 4th and 7th positions, which all end up falling on the same side of an alpha helix. So an alpha helix, as it's going along, is going to make 2 complete revolutions every 7 amino acids. And so 1, 4, and 7 end up roughly on the same side of the helix. That's the idea. All right. And so, 2 heptad repeats. And these are alpha helices, but they are amphipathic. So amphipathic means it's not hydrophobic, because that would be repels water. And it's not hydrophilic. That would be, attracts or is happy with water. It's amphipathic because it has one side that attracts water and one side that repels water. So you can think of it as two-faced. Yeah. And heptad repeat 1 and 2 are both these two-faced alpha helices. And we will come to see that that is really important because that's going to be what drives the fusion process. You'll also see some people call these the fusion motor, heptad repeat 1 and heptad repeat 2, because it actually supplies the energy to cause fusion to happen. Below HR2 is the membrane proximal external region, which is a highly conserved region of the protein that's just outside the transmembrane, which is the TM. And the MPER is going to have some very big, very hydrophobic amino acids. And we think this directly interacts with lipids. And it's basically going to splash some lipids from the virus, just like you do in the swimming pool, splash, splash, splash. It's going to splash some of those up toward the cell and actually create a little bridge and merge the cell membrane and the virus membrane. Yeah. When you make mutations down here, that is the process that no longer happens. All the other things work, but that part does not work. So, yeah. And then CT is the C-terminal region. That's like the tail that's going to hang down inside the virion of HIV. All right, so let's talk about some of these parts that we've just mentioned. So we said there was a furin cleavage site. Well, HIV-1 actually has 2 furin cleavage sites, because why not? Why not be extra horrible? A furin cleavage site is actually pretty easy to recognize. It's going to be a patch where 3 out of 5 or 4 out of 5 amino acids in that patch are going to be lysine or arginine. And lysine and arginine have these single letter amino acid codes K and R. So looking over here, can you spot them? Can you spot the furin cleavage site? Here's one, REKR. So RKR, yep, great. And the cleavage happens right after that arginine, the R. And here we have KRR. And wouldn't you know, right after that R, between the R and the V, there's another cut. So here you can see the cleavage sites right there and right there. The first cleavage is there, and if that gets cleaved, it helps the second cleavage to happen. It sort of frees it up, gives the enzymes a little better access. And each of these will be done by either furin or an enzyme like furin. There are a lot of enzymes that are similar to furin and will cut after these. And frankly, there are a lot of ways that this happens. Once this happens, it frees up the fusion peptide, which is the next thing. And so let's look at our fusion peptide. There you go. So this is a picture of HIV. And I should point out that the HIV envelope is a trimer because there are 3 copies of the envelope protein that together form that little shape. And so here you see a fusion protein coming out of this side. Here you see fusion proteins coming out of this side. And these dangly, wiggly things are different shapes that the fusion protein can take. And each of these has been found in a different study. The thing over here is it's naming a particular crystal structure of the protein with an antibody, which is the first part, and the fusion protein, which is the second part. So there you go. So yeah, and so this is the fusion protein dangling out, and yeah, guys, everybody, yeah, you don't want nobody wants to see the fusion protein of HIV dangling out. It's not something, yeah, that's good at all in any way. So keep that fusion protein tucked away, and then HIV can't infect, basically, yeah. All right, so fusion protein is there, and it looks like that. Heptad repeats, because why not? So here, this is a sort of a wheel diagram of the alpha helix. So it's a long alpha helix. It actually looks like a telephone cord, like so, on my old-fashioned work phone. And so if you were to look down the cord, which is to say, let's see if we can do it, this way. Oh, you can't see because my hand's in it. There you go, just like that. This is showing which amino acids are going to be on which side. And so the order they go, this is position 1, 2, 3, A, B, C, D, E, F, G. 1, 2, 3, 4, 5, 6, 7. And you can see that 1, 4, and 7 end up on the same side of the helix, just like we said. And so if the amino acid has a hydrophobic side chain, they've colored it in sort of dark. And if it has a hydrophilic side chain, they've colored it in light. And so you can see that this side is pretty darned hydrophobic. Yeah. And just a moment, let's turn the lights back on. There we go. Yes. And so heptad repeat 1 has a hydrophobic side and a hydrophilic side that's a little bigger. Heptad repeat 2 also has a little hydrophobic side and a much bigger hydrophilic side. And together, these two are actually going to come together. So when they get exposed during the fusion process, and again, you don't want to be exposing your heptad repeat 1 or your heptad repeat 2. It's not a good idea. But when that happens, these two things will then come together. And so the hydrophobic sides were nice and happy inside the envelope protein. But when they get exposed to the water, they get very unhappy in the sense that they are now bumping into water and they are hydrophobic. And so they will actually come together, just like that. And the two hydrophobic sides will end up on the middle of the sandwich. And yeah, that process of the hydrophobic sides basically retreating from the water and binding to each other is the force that is going to cause some of the fusion to happen. Okay, and I think this is the last part that we're going to look at in detail. So down toward the tail, remember we had that MPER. And here it is, membrane proximal, I don't remember what the ER stands for. You'll have it in your notes. This is going to actually be an amphipathic helix as well. And so it can sit in a membrane. And so this is from a different paper where they actually test that. And it has a hydrophobic side that is buried down in where the hydrophobic part of the membrane would be. And it has the hydrophilic side, which is up nestled amongst the head groups, which are charged in the membrane. So yeah, it is perfectly built to kind of float at the membrane-water interface. And this is the part that we think will actually end up carrying some lipid molecules from the virus membrane up to toward the cell membrane and actually creating that bridge, a process called hemifusion. And then underneath it, you can see the structure of the transmembrane, which, like we said, is an alpha helix of mostly hydrophobic amino acids. So there you go. All right, that's the end of this section, and we'll get in toward the mechanism in the next section.